Hello students, today you are going to study few points from the chapter that is plant water relation, and the point is first point of today's lecture is transpiration. Let us see what is transpiration. Plants absorb water constantly and continuously. Hardly 5% of the total water absorbed by roots that is utilized for cell expansion and the plant growth. Remaining 95% water becomes surplus, which is then lost into the atmosphere through its aerial parts like the leaves mainly. Hardly 1% of surplus water is lost in the form of liquid, and 99% surplus water is lost in the form of vapor. So the loss of water in the form of liquid is called as guttation. So I repeat the same sentence, that is the loss of water in the form of liquid is called as guttation, that is Guttation, guttation. It occurs through special structures called water stomata or the hydrothodes. The loss of water in the form of vapor is called as transpiration. Yes, this is most important thing. The loss of water in the form of vapor is called as transpiration. Through it occurs through the leaves, stem, flowers, and the fruits. Most of the transpiration occurs through the leaves. That is called as a foliar transpiration. The actual water loss during transpiration occurs through the three main sites. First site is cuticle, second one is stomata, and the lenticels. Third one is lenticels. Accordingly, three types of the transpiration are recognized. First one is cuticular, stomatal, and the third one is lenticular or the lenticular type of transpiration. So I repeat, there are three types of transpirations found in plant, that is the cuticular, stomatal, and the lenticular type of transpiration. Let us see one by one. Cuticular transpiration with the help of slide. Yes, cuticular transpiration. Cuticle is a layer of waxy substance, cutin, present on the outer surface of the epidermal cells of leaves and stem. Cuticular transpiration occurs by simple diffusion and the contributes about 8 to 10% of the total transpiration. Cuticular transpiration occurs throughout the day, and its rate is inversely proportional to the thickness of the cuticle. Let us see the yes, this is cuticular. Now just see the lenticular transpiration. Second type is lenticular transpiration. Let us see what is lenticular transpiration. Lenticles are small raised structures composed of loosely arranged complementary cells. Each lenticel is a porous tissue consisting of cells with large intracellular spaces in the periderm of the secondary thickness organs and the bark of woody stem and the roots of dicots. Lenticels are present in barks of the old stem and the pericarp of the woody fruits, but are absent in leaves. So these are only present in the woody woody fruits, and the barks of the old stems. So lenticular transpiration contributes only about 0.1 to 1% of the total transpiration. Okay, so rate of the lenticular transpiration is very slow. Okay, it is also occurs throughout the day. So these two types of transpirations mainly occur. That is the first one is lenticular, and the second one is the cuticular transpiration occur during the daytime. Let us move towards the most important transpiration. Before moving towards the last one, that is the stomatal transpiration, let us see. So this is nothing but the lenticular pores. These are lenticular pores. So transpiration occurs through these lenticular pores is called as the lenticular transpiration. Let us move towards one more transpiration, that is loss of water from the plants towards the external environment is called as a transpiration. So third type is stomatal transpiration. So you can see this type of transpiration with the help of the diagram. So before moving towards the actual description of the diagram, you must have some idea regarding the what is stomatal transpiration. So stomatal transpiration, uh, let us see what is stomatal transpiration. Stomata are minute apertures formed of two guard cells and the accessory cells. They are located on the epidermis of the young stem and the leaves. Leaves generally show more number of stomata on the lower surface. Depending upon distribution of stomata on the leaves are categorized into three types, namely epistomatic, means that when the stomata are present on the upper surface or the upper surface of the leaves are called as epistomatic. Then the hydrostomatic, hydrostomatic means when the stomata are located on the lower surface, then these types of leaves are called as the hydrostomatic leaves. And the ampistomatic means stomata, when stomata are present on both surface, that is upper surface and the lower surface, then that type of leaf is called as a ampistomatic type of leaf. This type of thing is mainly found in the so many grasses. Hypostomatic condition is mainly found in the xerophytes like the nerium, and the epistomatic type of leaves are mainly found in the lotus. Okay, so lotus leaves show this type of epistomatic stomata. Okay, so this is simply regarding or the in lotus leaves stomata are only located on the upper surface, and in nerium stomata are located on the mainly lower surface. While in case of grasses, stomata are mainly present on the upper as well as on the lower surface. Okay, so these are minute apertures. Stomata means these are small apertures located on the surface of the leaves. Upper surface, it may be the upper surface or maybe lower surface, and these minute apertures or the pores are guarded by the two guard cells. Okay, and those cells are called as guard cells. Okay, as far as dicot plants are concerned, dicot plants have uh, kidney-shaped guard cells, while the monocots have the dumbbell-shaped guard cells. So this is the basic difference between the dicotyledonous stomata and the monocotyledonous stomata. Let us see the stomata in the dicot leaf. Okay, so this is typical dicotyledonous stomata, and this stomata, this aperture is guarded by these two kidney-shaped guard cells. Okay, and this is minute pore through which transpiration process occurs. So this is simply regarding the typical structure of the dicotyledonous stomata. Let us move towards the one more point, that is the opening and closing of the stomata. This is most important point of the before moving towards the actual opening and the closing of the stomata. You must have some idea regarding the basic things about the stomata. In Cyprus, both kidney and double-shaped guard cells are present. Guard cells are living nucleated cells with unevenly thickened cell wall. Inner cell wall, that is cell wall is of guard cells is thick and the inelastic, and the lateral cell wall is thin and the elastic. Guard cells contain few chloroplasts which are capable of poor photosynthesis. So these guard cells have photosynthetic pigments, so they take over the function of photosynthesis also. Okay, but that photosynthesis process is very slow, so that type of photosynthesis is called as a poor photosynthesis. Guard cells have ability to change their size and form due to which stomata opens or the close. Stoma, a typical stoma is an elliptical pore. Stoma, what is stoma? Stoma is an elliptical pore formed due to the specific arrangement of guard cells. It is through the stoma, excess water is lost in the form of vapor. So, excess water is thrown outside the body of the plant into the external environment is called as transpiration, but that transpiration is in the form of the vapor. So, that water lost is in the form of the vapor. Okay, now just see accessory cells of the which are present in surrounding area of the stomata. These are specialized epidermal cells surrounding the guard cells. Their number is variable and are uh, reservoirs of the K plus ions. Reservoirs of the K plus ions. If we accessory cells are reservoirs of the K plus ions, and these are also called as subsidiary cells. These accessory cells are also called as subsidiary cells. So this is simply regarding the stomata which is present in the typical dipod plants, in which a stoma is guarded by two guard cells, and uh, these guard cells are further surrounded by epidermal cells or the accessory cells. Okay, so these guard cells also contain photosynthetic pigments, so they take over the function of photosynthesis, as I said earlier. Okay, but that photosynthesis process is very slow. That is also called as a poor photosynthesis. And the main thing is that accessory cells are specialized epidermal cells surrounding the guard cells. Okay, and their number is variable, and these are called as reservoirs of the K plus ions, and these are also called as a subsidiary cells. Okay, let us see regarding the opening and the closing of the stomata. Let us see the opening and closing of stoma. Opening and closing of stoma is controlled by turgor pressure of the guard cells. During daytime, guard cells become turgid due to the endoosmosis. Thus, turgor pressure is exerted on the thin cell wall of the guard cells. Being elastic and thin, lateral cell wall are stretched out due to the kidney and dumbbell shape. Inner thick wall are pulled apart to open of the stoma. During night time, guard cells become flaccid due to exoosmosis. Flaccidity closes the stoma almost completely. Endoosmosis and exoosmosis occur due to the diurnal, due to the exoosmosis and endoosmosis occur due to changes in osmotic potential of the guard cells. Different theories are proposed to explain the changes in the osmotic potential. According to the starch sugar interaction conversion theory, Stewart in 1964, during daytime, enzyme phosphorylase converts starch into the sugar, thus increasing the osmotic potential of guard cell, causing entry of water. Thereby, guard cells are stretched and stoma widens or the opens. So during daytime, due to this, stomata open, and the reverse reaction occurs at the night, bringing about the closure of stoma. So this can be represented by this particular, yes. So due to the phosphorylase enzyme, starch gets converted into the sugar, and due to this, turgor pressure increases and the stoma opens during daytime. And this process reverts to this daytime process, that is, in night sugar gets converted into the starch again. Okay, so this is simply regarding the opening and closing process, or this theory was proposed by Stewart in 1964. Let us move towards one more theory. According to the theory of proton transport, Leavitt in 1974, stomatal movement occurs due to the transport of protons, that is H plus and the K plus ions. During daytime, starch is converted into the malic acid. Malic acid dissociates to form the malate and the protons. Protons are transported to the subsidiary cells, and the K plus ions are imported from them. Potassium malate is formed that increases the osmolarity osmolarity and causes the endoosmosis. I repeat, according to this theory of proton transport, this theory was proposed by Leavitt in 1974. Stomatal movement occurs due to the transport of proton H plus and the K plus ions. During daytime, starch is converted into the malic acid. Malic acid dissociates to form the malate and the protons. Protons are transported to the subsidiary cells and H plus ions are imported from them. Potassium malate is formed that increases the osmolarity and causes the endoosmosis. Uptake of K plus ions is always accompanied with the Cl plus ions. Cl minus ions, sorry, accompanied with the Cl minus ions. At night, uptake of K plus and Cl minus ions is prevented by abscisic acid. I repeat, that acid is abscisic acid, changing the permeability of the guard cells. Due to this, guard cells become hypotonic and thereby become the flaccid. So this is simply regarding the second theory. This theory was proposed by Leavitt in 1974. In this theory, he reported that there are three ions actually responsible for the closing and the opening of the stomata. That is the H plus ions, K plus ions, and the Cl minus ions. Okay, so they play important role in the closing and the opening of the stomata. Okay, so this is second theory regarding the opening and the closing theory, and the first theory that was proposed by Stewart in 1964. Okay, before the theory of Leavitt. Okay, so this is simply regarding the two theories which are uh, explaining regarding the how stomata of the plants open and close. So this is simply regarding the opening and the closing of the stoma. Let us move towards the significance of this transpiration. First, see the advantages. First advantage is it removes the excess of water. Second one is it helps in the passive absorption of water and the minerals from the soil. Third one is it helps in the ascent of sap. Fourth advantage is as stomata are open, gases exchange required for photosynthesis and respirations is facilitated. Fourth one is it maintains turgor of the cells of the plants. And the fourth one is transpiration helps in reducing the temperature of leaf and in the imparting cooling effect. So these are different advantages of the opening and the closing of the stomata. Let us move towards the one more point, that is the disadvantages of the this particular opening and the closing of stomata. Excessive transpiration leads to the wilting and the injury in the plant. It may also lead to the death of the plant. So this is most important thing, and this is disadvantage of the opening and the closing of the stomata. Let us move towards the transpiration as a necessary evil. For stomatal transpiration to occur, stoma must remain open during daytime, and when stoma are open, then only the gases exchange needed for the transpiration, respiration and the photosynthesis will take place. If stomatal transpiration stops, it will directly affect productivity of plant through the loss of photosynthesis and respiration activity. Hence, for productivity, stomata must remain open. Consequently, respiration can not be avoided. Hence, Curtis in 1926 regarded transpiration as a necessary evil. Okay, so this is simply regarding the transpiration process is most important process, and it should be uh, properly done in the plants. Okay, so this is simply regarding, so in this way, in this chapter you have studied so many points, starting from in this chapter you have studied so many things like the properties of water. Okay, so you have studied properties of water. Then the most important thing is water absorbing organ, that is root. You have studied parts of roots or the regions of root, that is meristematic region, then the zone of elongation, then the root of root hair zone and the maturation zone. Then we have studied the structure of root hair. Okay, it is the elongation, that is the cytoplasmic extension. Root hair is nothing but the cytoplasmic extension of the root cell. Okay, then we have studied the water available to roots for absorption. Then we have studied the absorption of the water by the roots from the soil. In that we have studied imbibition, diffusion. Okay, then we have studied the osmosis also. That osmosis includes two types of the osmosis, that is the exoosmosis and the endoosmosis. And there are three types of the solutions, like the hypotonic solution, and the isotonic. I repeat, hypertonic, and the isotonic solution. Then we have studied the equation, that is the dPd is equal to OP minus WP, that is water potential. Then we have studied the water potential and the factor affecting the water absorption. Then we have studied the plasmolysis. Then the most important thing is that the path of water across the root, that is from the epiblema up to the xylem into the stellar region. Okay. Then we have studied the mechanism of absorption of water, that includes two types of absorptions, passive absorption and the active absorption. And that includes again the osmotic absorption and the non-osmotic absorption. Then we have studied the translocation of water. There are three theories proposed by three different scientists to prove the translocation of the water. That is the root pressure theory, that is also called as the Whittle theory, that was proposed by the Priestley. In the second theory, that is the capillarity theory, that is also called as a physical force theory. Okay, so that theory was proposed by Boehm in 1863. Then there are some objections or the limitations of the capillarity theory. Then there is one more theory, that is the cohesion tension theory. Okay, that was proposed by Dixon and Joly in 1894. Then we have studied the objections or the limitations of this particular theory, cohesion theory. Then we have studied one more point, that is the transport of mineral ions, transport of food material, path of translocation, vertical translocation. Then we have studied the mechanism of the sugar transport through the phloem. Then we have studied the transpiration in the plants. In that we have studied the cuticular transpiration, lenticular transpiration, then the stomatal transpiration. So we have studied all these points in the transpiration. Then the most important thing is that structure of the stomatal apparatus, or the structure of the typical dicot stomata. A typical dicot stomata consists of a middle aperture which is guarded by two guard cells, okay, kidney shaped guard cells, and these two guard cells are further guarded by the accessory cells or the subsidiary cells. Then we have studied the most important thing, that is the opening and the closing of the stoma, and this thing was proved by two scientists. This thing, that is opening and the closing of stoma, was studied by two different scientists, that is the Stewart and the Leavitt. So Stewart proposed his theory in 1964, okay, and Leavitt proposed his theory related to the opening and the closing of the stomata in 1974. Then we have studied the significance of the transpiration, that is under this we have studied the advantages and the disadvantages of the transpiration. I repeat, significance of transpiration. In this we have studied the advantages and the disadvantages of the transpiration, and at last we have studied the transpiration is a necessary evil. In this way you have studied all these points in this particular chapter. Now this chapter is over. This is chapter number 6, that is water, plant water relation. Thank you. If you have any doubt, you can ask me later on. Thank you.